said, I will focus today on antibiotics produced from Bacillus. So this context of these talks, as you understood, is related to antibiotic resistance. It is quite a serious problem, actually, and it is stated in many reports for the World Health Organization. So now we know all of these antibiotic resistance is due to the resistance to antibiotic drugs that was once able to treat these infections. This antibiotic resistance is new. It's not new in bacteria. In fact, it predates about before medical use, but it is related to the widespread and misuse of these antibiotics. And these generated strong selective pressure that yield to the emergence of these resistant strains. Situation is well known, as the previous talk highlighted, so I will skip all of these go very over it very quickly. And this means that in the future, these antibiotic resistances will grow and grow and will be in, sorry. Oh, yes, I'm going too fast. And it will be very serious in the future, this antibiotic resistance. However, there is, in that context, there is kind of paradox. Because in the past, there is a huge field of research in antibiotic discovery. And now it is decreasing more and more. But by contrast, there is a huge increase in antibiotics resistance. So this means that we need to discover new drugs and trying to find these new drugs from alternative producers. So in our lab, we were focusing as Bacillus as a source of new antibiotics, and we were focusing on peptidic antibiotics. You would say, why choosing Bacillus? Bacillus is sole bacteria, and it is also associated to the gut. It is used as a model organism for research, so it is well known in labs. And it's also used in biotech industry for protein production, meaning that we know very well its behavior in large-scale bioreactor. More importantly, the Bacillus genomes, more than 10%, is devoted to the synthesis of bioactive compounds. And today, I would like to show you 3 examples of such antibiotic compound. The first one will be related to what we call the antibiotic, which has ribosomal modified peptides. The second one will be related to bacteriocin that are also ribosomal peptides, but they are not modified. And finally, I will end with the lipopeptide, which are NLPS compounds. So what are antibiotics? Antibiotics are lanthionine containing peptide, and they present antimicrobial activity. They are synthesized on the ribosome, and they undergo post-sensory modification, such as the hydration of serine and threonine residue to form what we call dihydroalanine, DHA, and dihydrobutyrin, DHB. These DHA and DHB could undergo further modification, such as microtype addition onto neighboring cysteine residue, to lead to the formation of thioethyl bridges, yielding finally to the so-called lanthionine and methyllanthionine compound. In the field, lysine is the reference molecule that has been used worldwide as food preservative. Antibiotics are produced by Bacillus, as I said, but also by bacteria from the Lactococcus and Streptomyces genera. They are, most of the time, they are heat-stable peptides. You can even boil it for several hours without losing biological activity. They are small peptides also. They are usually less than 5 kD. This means it's quite difficult to purify from the process. They are toxic peptide that has been specifically designed to kill other gram-positive bacteria. So they are produced by gram-positive bacteria, and they are active against other gram-positive bacteria. And the mode of action is either to inhibit the peptidoglycan synthesis by binding on the lipid 2, which is this carrier of the peptidoglycan monomers, but also by inducing pore formation across the plasma membrane, again by targeting the lipid 2. In all cases, antibiotics are synthesized on the ribosome as a pre-peptide consisting of the leader peptide and a core peptide. This core peptide then after undergoes this postural modification that are encoded by 1 or 2 genes depending on the type of antibiotics. The last step is the removal of the leader sequence by some proteases. Other genes are also involved in the synthesis of the antibiotics. Transporter protein to have secretion of the antibiotic outside of the cells. We have also a two-component response regulator that will regulate the production of these antibiotics. And as I said, the producer strain is a gram-positive bacteria, and these antibiotics are active against gram-positive bacteria. This means that the producing strains must be outfitted with very efficient cell-protecting systems, and these protecting systems are encoded by the so-called immunity genes. In the lab, we were at that time focusing on the specific strains of Bacillus amyloidosis, and we found out in the genomes 800 base per fragment. It was during partial genome sequencing. And these 800 base per fragment showed a very high homology with modification genes of the antibiotic mersacidins found in Bacillus subtilis. So this opens the question, is that our strain is a producer of antibiotics? To answer this question, we first have to highlight the antimicrobial activity of the strains and its culture supernatant. We need to develop a production shame in bioreactor, but also to set up the purification process. And this then allow us to perform some biochemical and biological characterization of the peptide. From preliminary experiments, we knew that the antimicrobial activity in the supernatant was stable and was sensitive to proteosis. And that also this antimicrobial activity was mainly centered on gram-positive bacteria, and especially on pathogenic bacteria such as Staphylococcus aureus or Listeria monocytogenes. After having setting up this production and purification shame, we went on to biochemical characterization, first by mass spectrometry. We obtained these mass peaks, and these mass peaks allow us to say that we were facing a new compound not described to date in databases. We went also on the characterization of the genes involved in the bare synthesis of this amylolysine compound. The gene clusters contain 2 immunity genes, 2 recognition genes, 1 gene involved in transport, and 1 gene involved either in modification and in proteases, proteolytic cleavage of the leader sequence. And if we have a closer look on the structural sequence, we can see here that we have in red 29 amino acid leader sequence. And in the core peptide, we have here, depicted in green, a highly conserved protein that is known to interact with lipid 2. Then after, we wanted to link the genotypes to the phenotypes, so we disrupt the structural genes, and we analyze culture supernatant of the wild-type strain and the disrupted genes. You can see here, these peaks in the wild-type correspond to amylolysine, which is lost for the disrupted strains. And we also lost the biological activity against Listeria species with the disrupted strains. Meaning that the identified locus is indeed involved in amylolysine biosynthesis. The last step was to prove that this amylolysine was an antibiotic strictosensus. And we perform this by using Latin standard and amylosine hydrolysate and analyzing them by LCMS. And we can see here that in amylosine hydrolysate we have these peaks corresponding to the Latin standard, meaning that the amylosine could be considered as a real antibiotics. All these biochemical characterization led us to propose quite of structures of the compound, and now we are trying to sort this structure by NMR. Then we moved on the molecular characterization of the biological activity. We first show by thin layer chromatography that amylosine was able to find in vitro to lipid 2. That amylosine was also able to disrupt the membrane integrity by transmembranous electrical potential measurements. And we also demonstrate by very simple cell leakage experiments that amylosine was able to perform a pore into the cytoplasmic membranes. These highlight that the biological mode of action of amylosine is based both on the interaction with lipid 2 and therefore on peptidoglycan biosynthesis, but also on membrane pore formation. So we have a dual mode of action of this compound, which is not so common in this type of antibiotics. You could find further information if you want in this publication. So now let's move to the second example related to bacteriocin, which are, as I said, ribosomal unmodified peptides, and more precisely on subtilosin A. We were at that time interested in working with a bacillus strain BSP1, which is a gut associated. And we first tested the activity spectrum of cell-free supernatant of the BSP1 strains of bacteria isolated from swine intestinal content. And we can see here that the culture supernatant of the strains was able to inhibit the growth of some pathogens, such as Clostridium perfringens, and also on some Listeria strains. So then we went on the characterization of the antibiotic compound, again by Mailutov mass spectrometry. And this mass peak led us to highlight that the compound responsible for the biological activity was indeed the subtilosin A. So we went on the genome analysis. We found out in the genome of BSP1 strain there was a cluster of genes that is known to be involved in subtilosin A synthesis. To prove that it was the case in our strain, we disrupt the structural genes, the SBOA and SBOX genes. And again, we analyzed the culture supernatant of both the wild type and disrupt strain by HPLC. You can see here for the wild type strain, we have here this piece corresponding to the subtilosin A, which has disappeared in the mutant strains. And in the mutant strains, we also lost the biological activity. Finally, with this, we also determined that, again, the mode of action is to interfere with membrane integrity. So the last example I would like to present today is related to lipopeptide that are NRPS compound. NRPS stands for non-reducible peptide synthesis. As you may all know, normally peptides and proteins are synthesized on the ribosome starting from the mRNA. In the NRPS systems, situation is a little bit different. We have genes also, but these genes are coding for multi-osmotic complexes known as synthetases, and these synthetases are able to synthesize the amino acids and the peptide and modified peptide. These synthetases are multi-osmotic complexes that are encoded by very large operands. They are organized in different modules, and each module is responsible for an amino acid incorporation. These modules are related to adenylation, condensation, and thiolation. You could have also some specialized domains. So I have to speed up a little bit. So this adenylation domains is involved in the recognition and activation of the corresponding amino acids. Thiolation domains is involved in bringing all together this amino acid into the condensation domain where the peptidic bonds will be formed. First example I would like to show you is related to the effect of the NRPS compound on biofilm formation. So you all know what this biofilm is. And biofilm is the predominant lifestyle of microorganisms. They are associated with high tolerance to exogenous stresses, including antibiotic treatments. That means that we have now to find new antibiotics that are able to fight against bacteria even when they are associated in biofilms. Again, at that time we were interested again to work with some Bacillus ammiculaceae strains, NT1. We knew that this strain was able to synthesize different types of lipopeptide from the sulfactin, phenocin, and ethylene A family. This lipopeptide is composed of a cyclic peptide with fatty acid tails. And we highlight that this lipopeptide was able to inhibit the growth of some bacteria, including pathogenic bacteria, in planktonic cultures, but also in biofilms. Second example I would like to show you today is the case of microsubtilene. Microsubtilene is composed of a cyclic peptide with 7 amino acids and with various homologues of fatty acid chain lengths ranging from C15 to C17. And we have also different fatty acid isomers from linear iso and anti-iso. And we determined that the C17 anti-iso homologue was by far the most active biological compound. So we were facing two main goals. First, to decrease the heterogeneity of the production toward the C17 anti-iso homologues, to have a production of pure compounds in our bioreactor. And we needed also to increase the level of production, because the level of production in wild-type strain is very low. It's not sufficient to develop a robust and efficient process. So, how to decrease the heterogeneity of production? We were starting from facts from the literature, and it is known that the incorporation of the fatty acid by the synthetase is made from the predominant fatty acid present in the pool, intracellular pools. We know also that 80% of the membrane lipids are outnumbered fatty acids. And the roles of these lipids in the surface membrane is to maintain the membrane fluidity in regard to temperatures. This means that membrane fatty acid composition is temperature dependent. And it is known also for a long time that a lower temperature outnumbered anti-iso fatty acid increase in proportion in the intracellular pool. So our strategy was very simple, was to perform culture at lower temperature to favor the synthesis of these outnumbered fatty acids, anti-iso fatty acids, in the intracellular pools that will in turn favor the synthesis of the C17 anti-iso microsubtilenes. So we performed different culture at different temperature for wild-type strains, but also for constitutive mutant to get rid of any effect of gene expression. And you can see here, when you decrease the growth temperature, you increase the proportion of the C17 homologues, both for wild-type and the constitutive mutant. These experiments were confirmed later by other type of experiment. I don't have time to present them here. And surprisingly, at lower temperature, we could also observe a huge increase of the productivity of the microsubtilene. So we wanted at that time to determine why. This higher level of production is not due to higher expression level of the MIC operand, as illustrated by semi-quantitative RT-PCR experiment. These results were confirmed with using a Lagzi reporter construct. But in fact, the higher productivity at lower temperature is due to the higher catalytic efficiency of the synthetase, as shown here by these ATP peptide exchange experiments which is the mean to measure the adenylation activity of one of the subunits of the microsubtilin synthetase. How to increase the production yield? By simply changing the native promoter with the PXIL-A promoter. PXIL-A is the promoter of xylose isomerase, and it is induced in the presence of xylose. So first, we determine the regulation of this promoter in the producing strains, and we found out that the optimal concentration of xylose was 1%. We also needed to define a carbon sources to ensure cell growth before the xylose induction phase, because we would like first to have a two-phase system with a growth phase and then after a microsubtilin production phase. Then after, it turns out that we set up a medium composed of the historical medium for lipopeptide production, supplemented with sucrose and xylose. So next step was to construct the overproducing strains by exchanging the native promoter with the PXIL-A and XIL-A its regulator in the wild type strains, and to analyze the productivity of both strains. Wild type strains was grown in bioreactor in lambda medium at 37 degrees, while the overproducing mutants was grown also in the modified medium at 20 degrees. And you can see here that we obtain after 72 hours 10 milligram per liter for the wild type strain, and we obtain almost 1 gram per liter for the mutant strains only in 48 hours. The last step was to analyze to verify that the microsubtilin produced was indeed the C17 anti-iso. So we went on the purification of these lipopeptides. Characterization by Marlitthoff mass spectrometry showed that mainly the C70s homologs was produced. And two-dimension NMR highlight that it was indeed the anti-iso C70s microsubtilin that was produced. And finally, we confirmed that the biological mode of action of that lipopeptide was related again to pore formation in some of these pathogenic yeast. Through these few examples, I hope that now you are a little bit convinced that Bacillus could be used as a source of a very interesting antimicrobial compound. And I'll thank you for your attention.
Thank you very much for this nice talk. One question, with the lipopeptides often have the side effect that they are hemolytic. Have you tested your lipopeptides with this?
Yes, it is known that, for example, surfactants are hemolytics, and microsubtilin depends on the concentration. But for sure, this in the future application will be more related to skin application, not to injectable. Yes, yes, yes.
So, more questions? We have some time. Yes, sir?
Are the known mechanisms influence the antibiotics on the bacterial cells?
The known mechanism of
Effect of this substances on bacterial cells.
they are making pores into the surface membrane. Yes, yes. So it's targeting this lipid too, which is intermediate of the peptide glycan synthesis. Could block the synthesis of peptide glycan. This is one mode of action. And some of antibiotics have other mode of action is they are able to make pores. They associate with lipid too, and they are forming channels into the surface membrane. So some antibiotics have one of the other one mode of action, and such as amylase, they have both of them.
Do you think that for this material resistance in future is lower than the previous antibiotics? Do you think that the bacteria can be resistant again to this new material very quickly? Or
It is for antibiotics. There are some strains that are resistant to some antibiotics. But the main advantage is that these antibiotics, due to their structure, they are very difficult for bacteria to hydrolyze them, for example. They are very stable to proteolasis. I would say in natural conditions, because it depends on the dose, of course. Yes?
You have told us something about niacin as an antibiotic.
Uh-huh.
This is pH sensitive, so if you lower the pH,
Uh-huh.
it's highly doing its job, and if it's pH high, it's not working well. So you think your compounds are also pH sensitive?
No, we have tested this, and it is more stable on the right range of pH, and this is one advantage. And also we have made trials, for example, in poultry meat infected with Listeria, and niacin is more rapidly hydrolyzed by natural proteolases.
I suppose it may have synergistic effect with being using with the other antibiotic, yes?
I don't know, we never tested it, so I can't guess. Maybe it's possible.
Yeah, thank you very much. I have two questions.
Mm-hmm.
Maybe I didn't get you well. Looking at the title, it appears you have not identified a particular species you were working with. The title says antibiotic from bacillus.
Yes, because I present many examples from many specific species. That's why the title I would like remain more general.
Okay, you work with many species.
Yes, we work with Bacillus subtilis, Bacillus ameloquipacciens, and from different isolates.
Okay.
Because depending on the strain, for example, for lipopeptide, even in Bacillus subtilis, some of them are producing only iterines, some other produce 3 families of them. That depends on the strain.
We don't need them down with that. Number two is that why did you have to think of decreasing the heterogeneity of okay, thank you. Why did you have to think of decreasing the heterogeneity of the product?
First, if you want to develop drugs, you need to have few compounds. Okay? The first point. The second point is that the ability of the strain to produce this lipopeptide is fixed. And if you try to drive the production through the most active compounds, you will gain in efficiency. That's why. Because then after, if you want to purify it, all of these different homologues is very, very, very complicated. Because they are the only difference sometimes on the 1 carbons. Okay, this fact is quite. It is still possible to separate them, but if you want to separate isomers, iso or anti-iso, for example, it is almost impossible at large scale. That is why.
Okay, thank you.
Do we have time for one more question?
Thank you for your presentation. I would like to ask if we have some bacillus species are resistant to antibiotics, so could be fought by other species which produce antibiotics in your study, because I haven't seen any bacillus, just Clostridium and other species.
I don't know.
Because we have already in Egypt, we have isolated some genera, but we didn't identify the species from polluted water. It was really antibiotic resistant.
Okay.
This is for my question.
Okay. I have no idea about
Okay.
our strain is our resistant or not.
Okay, thank you.
I'm sorry.
So, thank you very much, and I want to thank all the keynote speakers of this morning.